Okay, so I think we'll start the class now. So could somebody please let me know whether the screen is visible, the slides are visible, and the and my voice is clearly audible or not? Anyone? Okay, thanks a lot. Okay, so again, I welcome you all to the course Bioinformatics Algorithms and Applications. So this will be the live tutorial session for the contents of week 6. So whatever Professor Pomiha has covered on the lectures for the week 6. So we'll try to see and solve some questions similar to and related to those concepts which came in the earlier iteration of the courses. And then throughout the session we'll just talk about some of the concepts that Professor Punia mentioned or went over in his class. Okay. And again, so this tutorial session will be taken by me. So I'm Sunandu Samanto, a PhD scholar from IIT Indus PSP department. And okay, so to recap, the last few classes or the last class, I think particularly what we covered. Um, so firstly, we talked about in the last class non-redundancy and uh, how to use different basically softwares or tools to improve or reduce the non-redundancy from a data set. We learned about CD heat. We talked about blast class a little bit, right? So those concepts are covered in the earlier classes. Some command lines. I think I also did like a small demonstration of how you can use CD hit, and then also in some way the concept of clustering came right because CD hit also uses the the concept of clustering to basically group different sort of sequences together to reduce the redundancy and so on, right? And we talked about k-means clustering, so basically identifying the firstly initializing the clustering process from different randomly selected points, and then identifying each data point to be a part of a particular cluster based on the distances, and then computing the averages, and then starting the process once again. Uh, and in the meanwhile, basically just uh, arranging or basically just computing the differences or the deviation within each clusters, and then ultimately trying to see in any further iterations whether that deviation, the sum total of the deviation improves or not. So that was kind of the part of what the concept of k-means clustering. So we did talk about that a little bit. We talked about the elbow method, how to find the optimal number of clusters. And I think we just talked about, another program, for example, Blast Class, where we can use or we can just provide some parameters for the Blast Class to use, and then at the same time we can use that to reduce the data set and so on, right? So those are some of the concepts, and then we talked about distances in the terms of Hamming distance and Euclidean distance, which can allow us to basically basically see if two sequences are in a close distance or in a close relation to one another or not, which can help us identify basically evolutionary connection or distance between multiple sequences. So those were some of the computations or calculations we did briefly, right? Uh, Aside from Hamming distance, we also talked about Euclidean distances. So basically, these are just amino acid composition based metrics to identify whether different sequences what basically will be the distance between different sequences, and then comparing two different distances you can kind of understand if they're closely related or not, right? So these are some of the concepts that we went over briefly, right? So another basically of the concepts that I think Professor Pumiho went over in the last class also, so it was related to information theory kind of thing, right? So GOR method is Garnier Osguthorpe Robson method. So this is one question, let's say, kind of relating to that particular method. So you can first try to solve this one, I guess, to begin the class. So here, what the question is asking is basically that if a protein contains a total of 1,000 residues, so out of the 1,000 residues, 600 of them are present in helix regions, right? Uh, and then the question asked about one particular residue, that is alanine, and it says that okay, among 100 alanines that are present in the entire sequence, 80 of them are part of helices, right? So the question is asking basically what is the information content of alanine in helix. So in relation to helix, you have to try to calculate the information content of this particular amino acid residue, which is alanine, right? And kind of the formula to basically calculate that would be something like this. So here, basically, you have to uh, understand or think about these different parameters. So these different parts. For example, here SSI is something that is mentioned. So SSI is basically secondary structure content at position I in the sequence, or secondary structure at position I in the sequence. Then X is any secondary structure. It could be helix strand, turn, coils, and then A could be amino acid residues, right? So to basically calculate these kind of things, I think I did not talk about this one in the last class. So you kind of have to calculate it in this way. Okay. So here it is asking first of all, so among the 800 residues, so here you see on the right hand side, so we have one subtraction, right? And here it is one term. We have to subtract this term from this one, from the left one, right? And then here this term is basically how you calculate is that so among 800 residues, if 80 are in helix, then this one, so probability of the secondary structure at position I, what this SSI is talking about, is equals to X. Okay, so equals to X means X is basically how many of that particular residue are present in helix. So you have to think of it in this particular this type of way. So among 800 residues, since 80 of them are present in helix, so you can write this term as 80 divided by 100, or basically 0.8, right? And then this one, so this sort of curly, curly symbol, so it basically indicates how many are not something. Okay, so here this divided by this P SSI equals then this curly symbol and then X pipe A. So this basically indicates that okay, among the alanine, the total of 800 alanine that we're talking about, how many of them are not present in helix. Okay, so this curly uh, symbol, that is basically indicating not present in helix here. Okay, so basically what we can do, we can then divide that by 20 by 100, because if 80 of them 80 of these alanine residues are present in helix, then of course 20 of them will not be present in helix. So we can get this term, the term that I will highlight right now. Let's just highlight it quickly. uh, okay, so this term, that the one that I'm underlining, so you can basically get this by doing ln 80 by 100 divided by 20 by 100. Okay, and then you have to do that minus, and then you have to calculate another term. Now here this term will consider not only alanine but for all of the residues that this question will be talking about. So similarly, this part in pink, in pink color, so it is basically talking about, so among all of the residues present, how many of them are part of helix. And then this part in blue, it is talking about among all of the residues in this sequence, how many of them are not present in helix. So this Cardi term is not, right? So basically you can do that again, minus ln, because 600 among 1000 residues are present in helix, so ln basically 600 divided by 1000 and divide that by total 400 by 1000. So you get this entire term, which if you calculate, you will get something like 0.98. So if something related to information theory kind of calculations are, let's say, given in exams or assignments, so you can kind of approach it in this way. Okay, so here the answer will be 0.98. So simple formula, you try to remember that, it might come in handy in the assignments and so on. Okay, so we talked about blast clust in the earlier classes, talked about CD hit. And then in this particular class, so Professor Grumiha went over several concepts. So firstly, relating to some of the protein, structure kind of concepts. So we talked about PDB and the different components of that, and then of course in the conversation came the concept of X-ray scattering, right? So X-ray diffraction or X-ray crystallography, that is basically the technique that is used by researchers to basically produce the crystal structures of certain proteins and biomolecules and so on, right? So here, this question, it is basically asking that attenuation of X-ray scattering or coherent neutron scattering caused by thermal motion is described by which of these terms? So is it Bragg's law, is it P factor, is it composition, or is it none of the above? So this particular term that we are talking about, it is also very important, and we'll see how this term is basically mentioned in the different structures, or kind of like introduced or in the different structures, or what kind of insights it can lend us into the structures. Okay, so we can try to answer this question. Maybe try to answer in the chat, and then we'll look at the answer. Okay. So in this class, we'll look into the PDB structures and the related concepts as well. Some of the statistical parameters that Professor Komiha mentioned, which can allow us to compare basically different structures, allow us to find out what sort of difference is between them, and just some concepts like that, contact maps, those kind of calculations, those things. Okay. So first we can try to answer this one. So try to answer in the chat, and then we'll look at it. So basically, here the answer will be P factor. Okay. Now P factor is a particularly important concept. We'll look at it, how it is written in a PDB file as well. Now what P factor basically is that it is, you can think of it kind of being constructed from this formula. So 8 pi square and then this average of U square, and then U in this case will mean displacement of a scattering center in angstrom, let's say, right? So basically, the concept that, for example, Professor Gomiha, how he introduced it, is basically that. So if we can hold an atom in one place in a rigid way, so you do not let it move, if that is possible, let's say, then we can ideally basically observe what is the ideal distribution of electrons around that atom, right? So in that case, of course, the images will be denser in the middle, and then it will be kind of falling off the farther you go from the nucleus, right? However, in real situations, so that does not happen, of course, and that is because the vibration of atoms, that sort of introduces an additional, you can say, additional fluctuating parameters that is present in these cases, right? So what basically happens is that the electron density is basically distributed much farther than this ideal situation, uh, and you can kind of think of it in a way that in the crystal lattice, whatever different molecules are present, so it sort of represents the different uh, molecules and their positioning in this crystal lattice, right? So basically, uh, it will include, so this PDB structure that you are ultimately getting, that will include the average of all of these small motions that are occurring for a particular atom. Now, that term that represents the motions or thermal fluctuations in this case, so that is basically called B factor. Okay. And the higher the B factor, basically you can think of it, the higher the fluctuation or motions in that for that atom is, and then of course the less significant the particular positioning for that motion will be captured basically in a way, right? So for example, Professor Bromia mentioned, so what could be the values that could allow us to compare different B factors which will allow us to compare the fluctuation levels for these different atoms. So for example, he said that under 10, you can think of it as a very rigid one. If it is greater than 50, then you can think of it as a very fluctuating atom in that way. So in molecular dynamics simulations also, so when you examine a system through like a dynamic compass, let's say, so throughout a particular time scale, you sort of try to investigate the fluctuations for let's say each amino acid residues or each atoms thereof. So in there also you can calculate the B factor and how it changes through time. And not only that, you can also calculate something, a statistical parameter like root mean square fluctuation, which will again allow you to basically compute the fluctuation level for different residues. So you can do that also, or you can calculate the B factors for particular atoms in particular residues also, which can give us some insights regarding the motion of these proteins that are present in the system, basically, right? On the other hand, Bragg's law, it is basically indicated by this one. So n lambda equals to 2d sine theta, and here you can see the scattering and the angle, that the theta angle that it is formed, and then the d sine theta, basically which distance it represents, and it is basically n lambda. So some sort of multiplier of lambda equals 2d sine theta. So that is kind of Bragg's law. Okay. So since we're talking about B factors here, right? So let me try to basically go to the PDB database, and then let us try to uh, understand or look into a structure of a PDB file, and then see what basically is inside. Okay. So here I will search for one PDB ID or PDB accession code, that is 3FQS. Okay. Now this 3FQS, what kind of protein it is? It is basically a kinase protein. So kinase proteins, I think I've talked about them in the past also. I think Professor Gomiha also mentioned some kinases in some classes like ES kinase, CS kinase, and so on. So these kinase proteins are basically like proteins that have an ATP binding pocket on one hand, and then have a substrate binding pocket on the very close to that ATP binding pocket, you can say. And then what it does is that upon ATP binding, so it transfers the gamma phosphate from that ATP to whatever substrate that is bound there. Usually some tyrosine, threonine, or serine residues of the substrate where the phosphate transfer will happen, and this basically allows different types of conformational changes in the substrate, which again comes in handy in progression of certain cell signaling, right? So kinases are very important in cell signaling, and you can find out many kinases whose structures have been submitted in the PDB database by many research groups, and among that we have opened up this one. This is 3FQS. So it is basically splint tyrosine kinase, a particular type of kinase which is important in you can say let's say immune cell related signaling processes. Okay. So it helps in, for example, B cell maturation, the B cell formation, and so on, and then the subsequent immune signaling processes. So it is kind of related to those. So what this PDB ID, basically when you open it up in the database, so you can see different values that are associated with them. So for example, you have something like resolution. So this is 2.10 angstrom. We'll talk about resolution also, I think, in some questions here. Then you have some R value that is mentioned here. Of course, as we saw, let's say in some earlier classes when we discussed about databases, so databases have to be highly annotated, highly integrated with other external servers and databases and so on, right? So here, for example, you see, excuse me. So here you basically see that a lot of external sources or resources have been linked within this PDB database. For example, the literature mentioned is here, so you can go through the DOI and then you can capture, whatever information is present there. Then the different chains and its related informations, the sequence related informations, and so on. Whatever other type of hetero kind of structures are present inside, for example, some ligands, etc. So that is mentioned here. The experimental data and what sort of different parameters obtained from that, those are mentioned here and so on, right? So if you basically go to these download files, so here you can download a lot of stuff. For example, you can download the structure, the 3D structure in PDB format, which we will look into. You can also download the sequences and so on. Also, at the same time, you can go to the display files and you can display the PDB format, what kind of information is mentioned there, and then you can also look into the FASTA sequence. So the PDB format, of course, it is very neat in terms of visualization, right? It has the coordinates and which we can use, different sort of visualization tools, for example, Chimera, VMD, PyMol, and whatnot. And you can visualize the structures very clearly, right? Which can allow us to investigate the structural changes and many different things in an efficient way. But not only that, the PDB format or the PDB file format, it is actually text readable and text editable. So you can use any sort of text editor to view the file and modify the file also. So here we will just look into the PDB format file in this display file section, and hopefully it is visible clearly. So if you open up a PDB structure through a text editor, so this is what we will basically see, right? So at the very upper section, you have a lot of informations which are part of this header, header parts, right, header components. You will see a lot of different informations which are present here. And at some point, you will notice that this sort of structured kind of columns, basically begin. So this is, you can say, one of the crux of the PDB files that we will use in different analysis and so on. So this thing basically contains the informations that we will try to investigate throughout different analysis. So first of all, on the very left column here, you will see it is written as atom, right? So here the term atom particularly indicates that it is a standard, for example, amino acid, standard atom that is part of an amino acid. So if this atom was from a different molecule, for example, a ligand, which is actually present in this PDF file, we can go to the very bottom part. We will see that those atoms are basically mentioned as het atoms, okay, heteroatoms. So these are not the atoms from a standard amino acid, so that is why they are indicated as het atoms, okay. Now, after atom, you see the numbering, for example, 1, 2, 3, 4, 5, 6, 7, which is present at the column to the right from this atom column. So can anybody try to guess what this numbering basically means? So this 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and so on. You see a lot of numbers in different columns. So what is the numbering at the second column indicate? What does it indicate? Anyone? So basically, the second column. So I see. Okay, I see one answer: AA residues, amino acid residues. Actually, it is not. So the second column basically indicates just adjacent to the atom column. So it basically indicates the atom number. So which number atom is that one? So that is what it is indicating. And then the column to the right, we see that N, Ca, Co, Cb, Cg1, Cg2, Nca, Co, and so on. So these are indicating what type of atom it is. Okay. Not only is it particularly nitrogen, carbon, oxygen, and so on. So not only that information, but what type of carbon it is, for example, right? So here, for example, we have the Ca. It is a C alpha carbon. For example, here we have C and then O. So that is the main chain carbon and oxygen. Here, for example, you have the Cb, Cg1, and Cg2. So these will be the beta carbon, these will be gamma carbon 1, this will be gamma carbon 2. So this type of nomenclature it is kind of following. Okay. And then towards the very right of that, we see VAL, TYR, etc. mentioned. So this is basically the 3-letter code indicating a particular amino acid. Right. So the first one is valine, the second one is tyrosine, the third one is leucine, and so on. And the column to the right, again, it mentions some number. For example, it starts at 363 and then it proceeds from there. So can anybody try to guess what this number indicates? So this 363, 364, and so on. So what is the number indicating basically? So here this column, this one is indicating the residue number or the amino acid residue number. So it starts from 363. Now for this particular protein, so you can think of it like a total of around 640 amino acid residues are present in this protein. So that would be like amino acid 1 to amino acid 640, around that. It's not exactly that, but around that. And this protein is basically composed of different kind of domains and so on, right? So there are two SH2 domains, two interdomain regions, and one kinase domain. This crystal particularly has captured the information about the kinase domain. So the kinase domain is starting from 363, and it is whatever is captured here in this case, it is kind of ending around 639. Okay, 639. So this is the structure for the kinase domain, and accordingly, the residue numbers have been included here. Okay. So after this column, so this 360, 364, 365, and so on. So on the right side, you see again different columns indicating different numbers. So now can somebody try to guess what this column indicates? So the one that I'm highlighting here. So 22.904, 21.687, and so on. So what can this one tell us about? Or if I slightly modify the question, so what does the next three column indicate together? So this next three columns, so 22.904 minus 1.375, 12.162. So those columns, they are kind of related in a sense. So what does those columns indicate together? Which is kind of one of the main parts of the information that is present here. It is, this one is used for your docking studies, MD simulation studies, everything. So these three columns together are basically indicating the position of that particular atom. So basically the X coordinate value, the Y coordinate value, the Z coordinate value. So the XYZ value, so basically the position of that atom in a 3D space, Cartesian space. So that is what is indicated by these three columns, basically, right? So of course, these three together will give us the point where this atom is residing. And when you consider all of the atoms that are sort of in this PDB file, so you will get basically the positioning of each atom, and then of course that means that each amino acid residues and their particular positioning and orientations and so on. So those informations you will get from these coordinate values, right? So right after that, again you will see one column where this 1.00 value is given. I think for majority of the atoms, this value is 1.00. So this is also one value that Professor Gumi had talked about. So what does this 1.00 value indicate, anyone? What does one column, that this column, that indicates here? So this is basically indicating occupancy. Okay. Uh, Now occupancy is basically, you can think of it in a way that if there are, for example, multiple positions of this particular atom that is captured through the process of X-ray crystallography, then you will see that the sum total of all of those positions will add up to be 1 in total. Okay. So basically, so for most atoms, you can see as it is in this PDB file, you will see that it is only 1. So that means that this atom is found for all the molecules that are present in the crystal at the exact same place. So that is why it is giving 1. Okay. Now let's say you are getting in some of the molecules in the crystal, you are getting it at a different position. And then for the majority, you are getting it for this particular position. So then you might see, for example, 2 lines that correspond to the same atom. So here, for example, for this particular N atom, you might have seen 2 lines that both indicate N. However, their occupancy, for example, in one might be 0.9, let's say, and then the other one will be 0.1, something like that. Of course, the coordinates will vary a little bit because the conformation of the atom positioning is slightly different. That will be the case in that case. So 0.9, 0.1, sum total to be 1. Basically, you might see something like that uh, for those cases. And for example, Professor Gomia mentioned this example. For example, like if a metal ion, let's say, binds to only half of the molecules in a crystal, then you can, let's say, indicate the occupancy for that atom to be 0.5 in the PDB structure. Okay. So it also relates to how many how many of these molecules that are present in the crystal you are seeing this particular atom. So it also relates to that as well. So that is the occupancy value. And on the right hand side, or on the right hand side column of the occupancy, so here one particular value is mentioned, for example 55.67 for the N atom, and for the subsequent atoms the values are also mentioned. So what is this value? Can somebody try to guess? So this is the value that we were initially talking about. If you look through all of the atoms, for all of those uh, atoms, these values are present, right? This value is present. So what is this value? So this column that is towards the right side of the occupancy column, so this is the B factor column, okay, the column for B factor. And you will see different values are of course present. So for the very terminal residues, for example, in this case, the terminal valine residue 363, you see a quite high value of B factor, right? 55.67, 55.47, 55.28, and so on. So it is over 50 kind of. So that basically indicates what? So this very high of a B factor value, what can this indicate? Similarly, let's say if you go to somewhere in the middle of this PDB file, you will see that, for example, in this 513, phenylalanine 513, you will see the atoms for that basically has a much lesser B factor, right? So 35.51, 35.16, and so on, as compared to 55, what you saw in the terminal valine, right? And then here again, more decreasing kind of values, for example, 29, right? So for the terminal valine, this high of a B factor, about 50 kind of, so that indicates that this residue is very highly flexible. So it has much thermal motion that was occurring, or that was captured in the X-ray crystallography, and that is the case for any kind of protein. But the terminal amino acid residues will be much flexible. If you examine the system through sort of a dynamic process, for example, MD simulation and so on, in that case also you will notice the same thing, that the terminal residues are very flexible. And on the other hand, the residues that are present in the core region, they tend to be less flexible, right? So this high flexibility is kind of indicated by the value of B factor, high B factor in this case. Okay. So that was kind of the PDB file. So we just talked about this, but what does this, for example, heat atom in this case indicate? So the atoms are indicating standard amino acid atoms, standard atoms that are part of amino acids, while these heat atoms will indicate any other molecules and its constituent atoms. So that is kind of indicating by this heat atom. So here this 585, that heat atom, so it is indicating a small molecule or a ligand that is present in this crystallographic structure. So it is basically a co-crystallized structure of this particular ligand, here indicated by 585 name, right? So that's kind of the overall architecture of a PDB file, at least its main coordinate portion. And then you will notice, of course, some crystallographic water is also present just below 585. These are the mentioned, or the information regarding the crystallographic water molecules. And then you will see at the very bottom, you will see some connect information. So these relate to different bond and connections and some information regarding those. So for example, if your for example, 16 residues, they are forming disulfide bonds. So they are at a very close proximity to form disulfide bonds. Then you will see some connect information regarding those cysteines sulfur atoms also. So those sort of linkage information is also present here. So if I ask, for example, one question. So here we have, let's say this arginine 434, right? I'm highlighting this one in the PDB file. So I guess this will be the entirety of the arginine 434, whatever is present here, right? So which atoms of this arginine 434 are part of the main chain of the protein? And conversely, which atoms are part of the side chain of the protein? You can indicate with the atom numbers that are present. For example, 525, 526, and so on. So can anyone try to guess? So which of these atoms are part of the main chain of the protein, and then which atoms from this are part of the side chain of the protein? Yeah. You can try to answer in the chat maybe. I think I see one answer. So CB, CG, CDNE side chains. Exactly. So in this particular case, for 434, so the main chain atoms are N, Ca, Co. Okay. So for every amino acid residue, that is the convention. So the N is basically talking about the main chain nitrogen atom. The Ca is of course the C alpha atom, and then the CO. These are the part of the basically the initial COOH that will eventually form the peptide bond, right? And this N is also part of that peptide bond. So in total, basically N, Ca, C, and O, these will be the main chain atoms. And the other ones, for example, C beta, C gamma, C delta, the Ne, CZ, NH1, NH2, and so on. So these will be part of the side chain of the amino acid residue. So these atoms are part of the side chain. And in many cases, for example, if you need to modify uh, the PDB file to include some sort of post translational modification and so on, since in some of those cases, for example, if you have a tool that can build a particular amino acid residue, if you only provide its main chain atoms and the corresponding three-letter code for those residues, uh, then what you can do, if you have some sort of tool like that, then you can what you can do is you can basically modify this PDF file, the text, whatever is present here. So let's say one thing you can do is that you keep the NCACO and then you delete all of the other side chain atoms, and then you keep whatever three-letter code for this amino acid should be, uh, and then you can try to command the software to build that particular amino acid residue for you. So for example, in Amber, which is a molecular dynamics software package, so there one sort of system preparation tool is present, which is called LEAP. So in that, what you can do is, if you, for example, keep for a particular atom only the main chain information, so NCACO, and then you delete all of the side chain information for that particular amino acid residue, and then you run the tlib command and ask it to build the system again. So it can basically, based on the three-letter code and then based on the main chain coordinates, it can try to place all of the side chain atoms in a position that makes sense. Okay, and then of course for actual before running any sort of simulations or dynamic processes, you have to minimize that structure because there might be some static clashes which will not be taken account by these sort of programs, right? So you need to minimize those build structures and so on. So we will look into, let's say, how to visualize the PDB structures. I think what I will do is that try to open up the PDB database, and then you can search for any sort of accession codes here. For example, just we did in the other case, for example, 3FQS, we can search for here, and then instead of displaying the structure through a text file, what we did earlier, so what we can also do is that we can download the files in PDB format, right? What we mentioned just earlier. So here, for example, we have this 3fqs.pdb open, downloaded, and we can open it up using some sort of 3D visualization tools. So I think Professor Grumiha in some classes, he mentioned about this tool PyMol, right? In this particular case, so what we will use is, let's say, one other alternative, which is called Chimera. It is UCSF Chimera. So developed from the research group of University of California, San Francisco. And then when you open up the structures, you will see immediately some interesting things. For example, some dotted lines are present in this region, right? Now, if you see in any PDB file that these sort of dotted structures are present, and then correspondingly, if you open up the sequence using the Chimera's favorite tab here, for example, here you can have the option of visualizing the sequence also. So this corresponding for the dotted structures that are present, corresponding to that, you will see some rectangular highlights in the sequence space also. Okay, so these are basically indicative of missing structures. So these are the amino acid residues whose coordinates were not captured in this particular dextrocrystallography. And then you are seeing a total of two similar dotted structures in this case. So those are basically missing structures, right? And as I said in that PDF file, when we opened up in text also, so what we saw was basically a co-crystallized ligand was present in the middle, named 585, which you can visualize here. So this is the co-crystallized ligand 585. So in this particular case, it is basically the active component of an FDA-approved drug for targeting this particular kinase. Okay, and then you will see that the entire basically secondary structures and so on are visualized uh, through this ribbon format here. Now for Chimera, there are many tools that are also integrated in its system. So basically, to create publication quality figures, you can go to this action tab, and then you can change the colors. You can change your visualization styles and so on using this preset. For example, if I go to preset and then if I let's say place something like this, so it will automatically change the background color, and then it will automatically give me some already available preset, right? And then you can color, for example, different molecules that are present or different parts of the secondary structures as well. So here, for example, this 585 molecule that is present in the binding pocket, this ATP binding pocket. So you can basically go to this action tab, then go to the color tab. You can open up more colors using these all options. And let's say I want to color the atom/bonds for this particular ligand molecule. Then I can select that, and then I can, for example, press magenta and then color by heteroatom. And I have right now a different sort of set of colors, right? Similarly, you can do uh, for the entirety of the protein secondary structure and so on. So here through the select tab, you can select basically different components. So first of all, you can select the entire chain, or if multiple chains are present, you can select one chain among those multiple chains. Then from chemistry element, you can select different atoms. So if group together, for example, all of the carbon or all of the hydrogen atoms you want to select, you can do that also. For the residue here, you can select particular amino acid residues or any other that are present here. For example, the crystallographic waters were present, the 585 ligand was present. And then throughout the structure tab also, you can select the backbones, protein, ligand, nucleic acid, and so on. Different options are available. So for example, if I select protein, and then if I go to action and then the color tab and open up all colors again, and then let's say I want to do coloring of this entire protein structure in light gray color. So I can select the ribbons, and then I can press light gray, and then it will change the color. Okay. So basically, that's how you can basically visualize whichever way you want to see the structure or to represent the structure for some publications. But there are different other analysis and also which are part of this the Chimera software and many other visualization softwares. You will also notice those kind of analysis. So for example, one very handy thing that you can do. So firstly, you can go to favorite and then use the command line. Okay. So we'll look at to some of the utilities of the command line also for Chimera. So whatever analysis you can do, for example, through this GUI, these different tabs and different options, many of those you can basically execute using the command line on the bottom side here also. So let's say if I want to select this ligand molecule 585, okay. So I can select, go to the structure and then go to ligand, and then I have selected this ligand molecule. Now let's say my interest was to find out which amino acid residues constitute the binding pocket for this ligand. Okay. Now I can of course move the cursor around these amino acids, and then I can see, okay, this is leucine 377. So that is probably because it is very close proximity to the ligand molecule, that is probably a part of the binding pockets and so on. But of course, that will not give us some exact answers if I'm looking for those. So one kind of a general approach to that can be, we can define an area or zone around the selected ligand molecule, and then you can try to see whichever amino acid is present there within 5 angstrom range, let's say. So that corresponds to the binding pocket. We can indicate something like that. Okay. So for that also, the zone selection option will also be available in this GUI. So the select option, you can go. First, you have to select one particular atom or set of atoms around which you want to define a zone. So here I have selected the ligand atom only. If you see this sort of green aura that is present around this ligand molecule, so I've already selected the ligand molecule, and then you can go to select, and then for example zone, and then it is asking us to select some sort of zone parameters, right? So here it is less than 5.0 angstrom from the currently selected atoms. So if I press this one as OK, so what it will do is that from the currently selected atom, which is the constitutive atoms of the ligand, it will define a 5 angstrom region, 5 angstrom radius, let's say 5 angstrom area just around that, and then it will select whatever amino acid residues are present there. So if I press OK, you will notice that some other part of the ribbon are also highlighted in green, right? So to more clearly see, you can open up the sequence. For example, from this favorite tab, you can open up the sequence, and then you will see certain amino acid residues are highlighted here. So basically, those amino acids which are highlighted are uh, part of the binding pocket, you can say, in a way, because they are kind of situated in a 5 angstrom distance from the ligand, right? So here you can hover over those single letter codes, and on the bottom it will give you which amino acid is that. For example, this one is leucine 377 of HN, then glycine 378 of HN, serine 379 of HN, and so on. Okay. So that is one thing you can do to kind of see what uh, amino acid residues kind of constitute the region of the binding pocket. You can try to visualize this, for example, again using these colors. Let's say I open up all options, and then I want to, let's say, give the ribbon something like a light blue type of color, and then let's say I visualize the side chain orientation of the different amino acid residues also, which I have selected. So I can go to action and then atoms and bonds, and then I can visualize the side chains. So you'll notice that some sort of uh, side chains are now visible in this part, right? I can maybe change the look of the side chain atoms. So I can go to that through this atom slash bond option, and then I can, let's say, do a ball and stick representation, right? So here the crystallographic waters are also selected. So I might want to delete that, for example. So I can go to select, I can go to residues, I can go to HOH, and then I can go to actions, atom slash bonds, and delete. So I don't need those crystallographic waters in this visualization. So I just delete those, right? And some other, for example, ASP 512. So here what is happening, I think, is not all atom information is sort of correctly being visualized here, but that's fine. So let's say I have highlighted this one now, and then one more thing which I might want to do is that let's say I want to see the surface representation. So for that, again, I can go to select, I can go to structure, I can select the entire protein, let's say, and then what I can do, I can go to actions and then surface, and then I can show the surface here, and this will again uh, allow us to see the entire system in slightly different way. So in the surface representation, it is interesting. We can also visualize what sort of cavity or cleft is forming in this binding pocket region, which is sort of making the space for the ligand molecule to bind there, right? So the surface representation is another interesting way to visualize. So what sort of surfaces you have present in the structure? So what could be the possible binding regions, binding sites, and so on? How deeply those binding regions are present in this molecule? How deep binding is occurring in this particular protein ligand interactions, and so on. So you can go through that through the surface representation. You can do another thing. For example, if you have created the surface, you can set the transparency for the surface also. For example, if you do it 30% transparent, then you will see some internal structure or the ribbon structure uh, that was visible earlier. So you can see some of that kind of starting to get visible. So if I set the transparency a bit higher for the surface, uh, let's say 70, right? So now you have kind of a combined sort of view. So where you have these ribbon structures also visible, and then at the same time you have the surface view is also there, right? And this could be like an interesting representation of the system that you're trying to investigate. You can use many of these commands through this command line also. For example, let's say this one is, so this particular structure is indicated by #0. If you notice here, the #0 is mentioned, right? So if I open up, for example, multiple structures at the same time, the first one will be #0, the second one will be #1, the third one will be #2, and so on. So for example, I can set the transparency from the command line also. So I can type maybe something like transparency. Hopefully I am doing it right. And then hashtag one. So it is saying error in hashtag one target values must be. This is not working here. So here, for example, I can use this transparency command. So you can look up different commands for more efficient usage of Chimera. It should be present in the Chimera documentations and so on, right? So I can use this transparency command, and then after that I can give, for example, this some sort of value for this transparency, and then after that I can indicate, for example, the residue range, and then I can give whatever kind of target I am uh, indicating through this transparency command. So it could be surface, it could be C for cartoon, it could be F for atom, and so on. So if I use 0 for this one, so now what you see uh, is that the whole surface transparency is becoming 0. So it is entirely visible. I can manipulate it in this way, and different kind of representation. Okay. Then I think the internal structure was not visible also, so I can maybe change this to target C, and then I can use 0 here. So you can try to basically use different kind of comments and to properly visualize this structure and so on. Okay. So I think the internal structure is visible here because I didn't select this part of the protein. So I can use 70 or something like that. So it gives us this kind of view. So that is basically how you can manipulate and view the overall structure for these PDB files or the protein files. Along with Chimera, if you go to these tool options, you will see the usage of different other parameters or other analysis. For example, here we have in the structure analysis portion, we'll see for finding H bond, finding distances, finding angles, torsions, and so on. So you can go through that. You can try to find surface analysis, for example. Some other options are there. So in the structure editing, you can try to add hydrogen, add charge, which could be useful in preparing your system for different docking processes. So as we saw in our structure also, there were uh, some missing regions, right? So you can model those, define or model slash define those missing regions, which we will see in some later classes when we talk about the homology modeling sections, right? So it basically uses for this, if you want to do it through this Chimera. So there is a modeler option that is integrated through this Chimera. So you have to download the modeler software separately if you want to use a local computer, or if the web server for modeler is available, then you can try to submit the job from there as well. Okay. And then there is other, for example, MD and sample analysis. So if you have a molecular dynamics trajectory, uh, so you can load that up, and then you can try to see in a form of movie kind of thing uh, what is basically the dynamic change that is occurring throughout this simulation time. So you can do different types of analysis here, right? Select clear. Uh, so we just talked about, for example, if you see one more example, so we talked about the distances, right? So we talked about that we can find out the distances between different atoms, let's say in this particular protein. So let's try to do one maybe. So here this lysine 402 is present. So if I just select that, if I go to the actions atom, just go and you just show this lysine, let's say through a ball and stick model, and then we let's say color it in red or something like that, or let's do it this way. So this is on lysine atom, let's say, the lysine amino acid residue. And then let's say there is this GLU 420 amino acid residue that is present, right? So we can even try to visualize it. We can just try to color it, let's say, in a different color, maybe, or same, and then by heteroatoms. So what happens is that in a lot of the kinase structures, so in the beta 3 sheet, which in this case will be this one. So this is beta 1, beta 2, and this is beta 3. So it has sort of a conserved lysine amino acid, which sort of forms a salt bridge like structure through this different helix. Okay. So glutamic acid from this helix, which has a name, it is called the alpha C helix. So that glutamic acid from the alpha C helix, it sort of forms a salt bridge like structure with this lysine. Now you can try to see, okay, so if it is forming a salt bridge kind of structure, so it should have, for example, a distance that is less than or around like 4 angstrom or something, right? So you can try to investigate that also. For example, if we take this CD atom, let's say for this lysine lysine amino acid, you can see lysine 402.A, and then after the CD is mentioned. So that is the delta carbon atom for this lysine amino acid. And then we, let's say, try to find out the distance between that and this CD for this one maybe. So this is the GLU or the glutamic acid CD 420. So you can try to find out the distances between them, and then we can try to see whether there is any sort of salt is present or not. I guess a better one would be if we try to just calculate instead of carbon, we can just calculate the distance between this NZ and then this CD. So you can do it in two different ways. So you can firstly, let's say, hover over this NZ atom. You can press Control on your keyboard, and then you can select this one. And then to select simultaneously this CD atom also, you can press Control Shift on your keyboard and then select this CD atom. So it will select both of these atoms, and then you can manually go to the tools and then the structure slash structure, I think structure analysis, and then the distance tab. So you can open that up, and if you have selected these two atoms, then you can create a distance between those. And then it is visible in yellow, so I can modify the color also, let's say, in some different way. So I think the distance is now visible. It is around 3.799, so it is within salt bridge forming range, right? So you can do that. Alternatively, you can, let's say I remove this and I want to use the command line. Again, same thing you can do. You can just give the distance command, and then you can mention whichever atoms you want to create the distance between. For example, this is the NZ of 402. So you can indicate the amino acid residues with a colon. So you can say colon 402. Then for the atom, you can press @ and you can press NZ. For the other one, for example, this is 420 glutamic acid. So the residue identifier will be 420 @ CD, because this is C delta atom. So this residue identifier, let's say it is 420 @CD. And if you press the enter here, I will find out that again the distance is visible. So through the use of command line, through the use of GUI, whatever tabs are present on the upper hand side, you can basically utilize those to analyze your structure in different way. For example, if you wanted to visualize some sort of structural features like let's say some electrostatic interactions are occurring or not between different residues, you can try to calculate the distances. You can try to model the loops using Chimera. You can visualize the surfaces and the binding dynamics, through an empty trajectory. You can do that also. So these different features are available in many other software suites also, for example, in Pymol, VMD, and so on. So those are also, those can also allow you to do this different, same kind of analysis basically. Okay, so that was kind of a brief uh, about visualizing PDB structures and then using Chimera to do some analysis. So I'll just do one thing. share the other screen and then we'll continue with the questions and so on. Okay. Okay. So let's say in a very related concept that we just discussed. So an atom with a temperature factor of less than 20. So what kind of atom it will be? So what is temperature factor that is mentioned here? Any idea? So this temperature factor, have we heard about this term or is it a new term? Yeah, it is B factor basically, right? So temperature factor is basically B factor. It's just another term for that. And what we learned is that, let's say, the atoms have a B factor of a very low value, then it is basically much rigid, right? So I think Professor Plumia, for example, introduced some sort of cutoff thresholds, like as some examples. So where he mentioned that above 50 B factor, so that will be very flexible. It has high thermal fluctuations that is captured, and then let's say less than 10, so it will be very rigid, so kind of fixed in a particular position. You can think of it in that way. So here for less than 20, you can basically say it is rigid. You can also maybe say located in a fixed position, kind of. That's sort of very similar. But 20 is, if that was less than 10 or 5 or very low value, then you could have probably said that. But anyway, for less than 20, you can generalize it by saying that it is a very rigid atom, not much thermal fluctuation that is captured in this particular atom. Okay. If it was, for example, something like 75, 78, that would be very high. So even around like 60 or something like that, so that will be very flexible atom. So we saw in our earlier PDB file, so the terminal valine 360, for those atoms that are part of the valine residue, the terminal valine, so we saw around like 55, 54, those kind of p-factor values. So those terminal atoms for the valine residue, that was very flexible. Those are very flexible, right? So high thermal fluctuation for those ones. So now again, another question we have is that, so among these following amino acids, let's say which one could be having the least propensity to be a buried residue? Now, what is buried residue in this case? So buried residue is basically residue that is or that are present in the core part of the protein structure, which is away from the surface, basically away from the solvent. Okay, so it is not solvent exposed in our cells, right? So which of these residues could be having the least propensity to be a buried residue? So again, some concepts of hydrophobicity and polarity will come in this question. So you can try to think of it in those terms also. So try to think in the line of, if an amino acid is buried, so of course that means that it is away from the surface, right? So surface is basically constantly in contact with the solvent or the water molecule surrounding the protein, right? So what sort of characteristic or features does this amino acid need to have if it is a buried residue versus a surface dwelling one? Yeah, I see. I think one answer in the chat: hydrophobic amino acid, right? So basically, you can compare the hydrophobicity uh, for these amino acids, and then you can say the most hydrophobic one will probably be uh, the buried residues, right? Now, in this case, it is asking the opposite question, which is which one has the least propensity to be a buried residue. So it is basically asking which one of these will be the most polar in that sense, right? So which one could be the most polar among these? So here, of course, we have a charged amino acid, if you remember. So this is lysine, right? And in this case, the answer will be lysine. So lysine being a charged amino acid, it is highly polar. So it will have the least propensity to be a buried residue. So you can kind of look into this chart also. If we remember, we go back to a couple of some classes earlier. So we have introduced this concept called hydropathy index, which is kind of like an indicator in a way, just a quantitative indicator of how uh, polar or how hydrophobic an amino acid could be. So basically, it was the favorability in terms of the transfer of that amino acid from a non-polar to water mediated area kind of thing, right? So from non-polar to polar solvent, if this amino acid is transferred, what will be the free energy of what will be the free energy difference basically? Uh, so the negative value will indicate that it will become more stable because it needs to liberate some energy to the outside environment for the final minus initial value to be negative. So that means from non-polar solvent to polar solvent transfer, it will be favorable by the residues that have the higher negative values, right? And in this case, you see, if you go to the lysine's corresponding value, you will see minus 3.9. So it is very highly uh, favorable in terms of transfer to a polar solvent. So lysine is, of course, a polar charged amino acid, right? Positively charged R group having amino acid. So it will be the least propensity among these. If you compare for the other ones, for example, valine has 4.2, and then methionine has 1.9, and then isoleucine has 4.5. So these are all non-polar aliphatic R group containing amino acids. So those will be more propensity. Those will have more propensity towards being buried residues, right? And again, if you look at this graph, which is mentioned here, so it basically compares the propensity at surface versus the propensity to be in a buried area. So surface propensity is mentioned by this blue color, buried propensity is mentioned by this red color. And if you see for lysine, this blue bar is quite high as compared to this red bar. So it has very high chance of being at the surface, basically, right? So these kind of graphs you can also refer to if you want to get a clear visual representation of the buried versus at surface propensity for these amino acids. Okay. So while talking about the structures and the comparison of different structures that you can get from the PDB database, let's say, so one statistical parameter that Professor Gomiha also mentioned. So it is a very important one, uh, and it can describe the deviation of certain structures from a reference structure. So if you have a reference structure, and let's say through some procedure you get uh, a different look at that structure, maybe, or maybe through a dynamic process for the same protein, you are getting a different structure. Different structure means the conformations for the constitutive amino acids will be slightly different, right? Conformations means their side chain orientations or the backbone for structure formation, the loop formations, for example, those structures can also differ in certain way. So those sort of deviations, which are conformation deviations, so which statistical parameter can allow us to get an estimate of that or a measure of that? So does anybody have an idea? So which statistical parameter can get us an estimate of uh, the conformational differences or conformational deviations? So I see one answer in the chat, standard deviations. So standard deviations will tell you about uh, basically the range or fluctuations of a particular value, right? So if you have any for certain data, the mean and the standard deviation, then you can get, for example, some idea about where this particular value resides in which range, right? For this one, the conformational deviation, it is also a deviation parameter basically, which will help us to get the difference between the structures, and that has a very particular name. So it is called root mean square deviation, or RMSD. So you might have heard of these RMSD values. It is basically incorporated in in a lot of, let's say, computational analysis where molecular dynamic simulations are involved. So to see the convergence of the simulations, whether the systems has attained a stability or not. So for that, the RMST value is used in a lot of sense. Then to see the structural deviations for certain regions of a protein, you can utilize these RMST values. To see, for example, the side chain orientation of a particular amino acid residue or some particular ligand molecules. Again, for those residues or ligand molecules, you can also calculate the RMST and you can see their conformational change. So the formula for calculating the RMST value is kind of like this. So don't worry about the formula too much. So it is just for the just for a like demonstration kind of thing. So how these values are calculated. So basically from I equals 1 to n. So for all the atoms that are constituting this particular protein, so we have to do a summation for all of those atoms. And then inside what we have is that mi, for example, here. So that will be the mass for this particular atom I. And then after that, you have something like xi minus yi whole square, right? So square of xi minus yi. Now this one is basically the square of the coordinate points or the coordinate values of whichever structure or whichever atom that you are calculating for, minus the same value, the same coordinate value for the reference atom, let's say. Here, y you can think of it as a reference point. Now, of course, if you are trying to calculate the RMSE for a 3D structure, then you have to calculate it for x, y, z coordinates and so on. And then since it is root mean square deviation, so basically what you do is that you square this term, this deviation value, and then ultimately at the end, when you kind of do the average, you just take a square root of this entire term. So that will give you the root mean square deviation. Now this one particularly, it is a mass weighted RMST. So that is why this mi and then its total m term that you are getting here. So that is because it is a mass weighted RMST. Now if you just calculate this non-mass weighted or the standard RMST, then what will happen is that so in that case, for all of the mi combined, that will give you kind of the total capital M here. So these terms will basically get crossed out in a way. So ultimately we'll be left by, so this will get crossed out in the meaning that let's say for each particular individual atom, if you consider the mass to be same, so let's say total n number of atom you have, so the total on the upper hand side you will also get n times small m, and then on the bottom hand side or the here instead of the capital M, so you can also rewrite that as n times small m, right? So in that sense, the small m and small m on here will be crossed out, and then you will be left by just the total number of atoms that are present in the system. That sort of way. So it will kind of be sort of an average in a kind of sense, right? And ultimately, you have to take the root over of that in the end. So that will allow you to calculate the RMST or the root mean square deviation. If we just take a quick look again. So I'll just use the AnyDesk this time. So if we just look at this file, let's say. So here I have some PDB files. Okay. Now if you notice, here we have something called structure 1, structure 2, structure 3, structure 4. So these are basically 4 structures that I have extracted from a molecular dynamics simulations file or trajectory. So I can open all of them up using Chimera. So I can open, for example, struct 1.pdb, and then at the same time I can open the structure 2, the structure 3, and the structure 4 in that order. If I press enter here, so you'll notice that Chimera is basically opening all of them at once. Okay. Now here, let's say I again to the preset that I did earlier. I think it was this one, right? So you will see that there seems to be some sort of the structure seems to be kind of mangled up in a way, right? Now, what you can do is that you can using the matchmaker tool of Chimera, you can basically align all of these structures. Just a moment. So here in this tools option, you will find for Chimera there is something called structure comparison tab. Then there will be something called matchmaker. Now through this matchmaker, what you can do is that you can align the different structures in with respect to a reference structure. For example, here on the this side, if you have structure 0 or hashtag 0 or structure 1, which I opened up first. So these hashtag 0, if you take that as reference structure, and let's say align structure 2, then you can basically press OK, keeping everything default, and you will see that in this Chimera terminal, so the two structures were kind of aligned. And at the same time, if you go to this reply log for Chimera, you will notice some sort of information regarding those alignments. So this matchmaker, apart from the alignments, the sequence alignment score was 417.9. Then it used, for example, these other parameters, this Needleman Wunsch, Blossom 62, so on and so forth. And then after that, you will see RMST between 160 pruned atom pairs is 0.813 angstrom, across all is 4.090. So that will give you basically the RMST values for those. So in this case, across all of the 277 pairs, if you consider the RMST, so it is 4.090. So the structures are quite different in comparison to one another. Basically, structure 2 and structure 1, which I have aligned together now. So let's say I want to align the remaining structures with the same reference structure, which was structure 1 or hashtag 0. So I can just use the match command again. So this is the command line alternative to the matchmaker tool here. So I can use, for example, hashtag. So I have aligned 1 with 0. So I can align hashtag 2 with the reference hashtag 0. You will see some change here. And I think those are all the structures. No, there is one additional. So I can use hashtag 3 and align that with hashtag 0 again. And then now you'll notice that all of these structures are kind of aligned together, right? And when you basically perform the individual alignments, you would have noticed in the reply log the particular RMST for those two structures were also indicated. For example, here we have 4.576, 3.796 angstrom, and so on. So that way, this way you can basically align different structures for the same system. If you get different ones from the molecular dynamics trajectory, you can align those and you can get the RMST values also. But in a trajectory file, so if you do the trajectory analysis in standard sense, you can basically get the RMST value for all of the frames that are present in this trajectory. So for example, in this one, here I have this RMST underscore back dot dat file, and this is basically from a 200 nanosecond molecular dynamics simulation. So for 200 nanosecond time scale, I have the trajectory information. On the left hand side, it says the frame number. Frame is the snapshot numbers that I have captured for this total simulation. And on the right hand side, we have the RMST values with respect to a reference structure. In this case, the reference structure was the initial structure. So whatever structure I used to simulate, that initial structure, the initial input, with reference to that, I have calculated the RMST. And this is basically this file basically contains for all the 20,000 frames that are part of this 200 nanosecond simulations that we captured here. So for all these 20,000 snapshots or frames, we have a corresponding RMST value with respect to the initial confirmation. And we can basically visualize it through some plotting techniques. For example, I can use, let's say the simple one, in this case, GNU plot. I can basically create a line plot for this RMST underscore back dot that file. I can visualize it, and it will give us something like this one. So on the x-axis, you will see the total frame numbers are mentioned. So in this particular case is basically if you divide the frame number by 100, you will get the time in nanosecond for which we have simulated the system. And on the y-axis, you will see the RMSD value, which is corresponding to the initial structure as reference. And this, the entire thing is basically giving you sort of a time evolution of this RMSD value. So basically, as function of time, you are plotting the RMSD values, how it is changing throughout the simulation. You will notice that at the very beginning, throughout the initial part of the simulations, so the value was kind of fluctuating in a way, right? Then it kind of got stable. Then here some slight fluctuation was there around the 16,000 frame number. And then after that, it seemed to stabilize again in this region, right? So when, let's say, throughout a decent simulation time, if your RMSD value is kind of getting stable, like what we have at the end, let's say it was stable like that, so that would indicate that your simulation had converged essentially. So RMSD you can use not only as an indicator of structural divergence or structural deviation, but also you can use it to analyze your simulation convergence in a way. Okay, so it is a part of that. It can be a part of that analysis also. Of course, you can just plot this file in different ways. For example, uh, let's say I have just, for example, simple. I have this kernel density estimation plot script. So very simple one. So here basically we will use the kernel density estimation method using Python. Okay, and we'll just plot that. So it is a part of this SNS, which is the Seaborn library in Python. And you can plot that and try to see what sort of output we get. Let's say very quickly, if we, let's say, plot that and generate the image, we will see that the distribution of RMSD was something like this. Okay, so here we are very clearly seeing a peak around this position. So maximum number of RMSD values that we have for these different frames, so those are kind of present at this position. Okay, so around this 2.4, 2.3, 2.4 kind of value, right? Around this part, maximum number of those values are kind of concentrated together, RMSD, right? For those frames. And then we see some small hump on the left hand side. So this could be the fluctuation that we saw in the time series. So in this particular case, we can say that, okay, this is the prominent RMSD value for this system. Okay. So you can use different methods to analyze your data, your RMSD data also in the case of trajectory analysis. So I will just move to the next set of concepts, I guess. Now that we have an idea regarding RMSD and so on. So let's try to find out the answers to some of the other questions here. So for example, here again, another question related to uh, the solvent accessibility and corresponding the bodied and surface propensity. So those kind of related terms. So it is a question related to those. So now here you have been asked that you have been given for certain residues the solvent accessibility for those residues, and then you are asked, okay, what which of those could be present at the interior part of the protein. Now it is very similar. uh, so at the interior part of the protein, basically at the core region, if you say, so in those parts, so mainly hydrophobic residues will be there, and those will be located at the uh, so where the basically solvent cannot reach those residues, because since they are mainly hydrophobic in nature, so they will not be favored for interaction with water and so on. So that is why they will be present in a buried location or at the interior part of the proteins, right? On the other hand, if you have some polar residues, some charged residues, so those will show more propensity towards being present at the surface of the protein, and those will more likely try to form some interaction with the surrounding water molecule or surrounding solvent. So for the uh, molecules, for the amino acid residues that are exposed to solvent or that are surface that are present at the surface, so of course the solvent accessibility means that is in angstrom square and area. So what area of those residues are accessible by the solvent, so that value will be very high, right? Conversely, if you see the amino acid residues that are present at the interior part, the inside part of a protein, if you say, if you will, so those will have a solvent accessibility of much lower angstrom square value, right? So in this case, if you notice the values, so I also see some answer A in the chat. So yeah, so basically, since alanine 7 has only the solvent accessibility of 0.2 angstrom square, and then valine 9 it has 1.2 angstrom square. So you can expect that those residues are in a pretty buried location where they are not exposed to solvent. So the interior part of the protein, those are present, right? So you can correspond these sort of solvent accessibility values with the location of those residues in the protein also. Now, related to PDB structures, we noticed some parameters were also present. So when we opened up the RCSP PDB and we typed the earlier PDB accession code 3 at QS, so you would have noticed that a lot of associated parameters were also present here. Among those, something called resolution was mentioned, right? Now, if you are asked, for example, for the same protein, 4 different research groups have submitted 4 different PDB structures, right? Now you have the different resolutions, for example, 1.75 angstrom, 3.43 angstrom, 1.14 angstrom, and 2.57 angstrom. Now, if you are asked to arrange the quality of the 3D structures from highest to the lowest based on the resolution, so can anyone mention in the chat maybe what could be the order of arrangement here? So I see, I think one answer C, A, D, B. So C, after that A, after that D, after that B. So here we are trying to get the order from the highest to lowest. Okay. Now for the highest to lowest, if you take the resolution, so in this case, what you have to remember is that, so this resolution values that are mentioned here in angstrom, so it is kind of in the same way as optical resolution. So it is the ability to distinguish between two very closely situated points, you can say, in a way, right? So that was the optical resolution. And in this particular case also, the lowest the value will be for basically the lowest the value will be for this resolution in angstrom values, right? So that means that you can distinguish between, let's say, two different points that are situated at that distance, right? So that means if it has a very high value, for example, 3 angstrom or 4 angstrom or something like that, then in this structure, if two points are present at a distance of, let's say, 1 angstrom, then that distinguishment is not that clear. So basically, the lower the resolution values, associated values for the resolution that are mentioned here, the higher the quality of the structure or the higher the actual resolution of the structure will be. So the lower the value here, the better the resolution for that structure will be, the higher quality that that structure will be. So in this case, the lowest value was 1.14 angstrom. So that will be the best quality structure, let's say, in terms of resolution. Then after that, 1.75 will come. Then after that, the D structure 2.57. After that, B 3.43. With the highest value here, so that will be the lowest quality or the lowest resolution structure. Okay, so that way you can think of resolution for PDB structures. So it is basically, as mentioned here, it is basically a metric reflecting the quality of the collected data on the crystal containing proteins or nucleic acids, anything, right? So in a perfectly aligned crystal, the proteins will share identical orientation, and then high resolution data can be captured in the diffraction pattern. Now, if the proteins within the crystal, they exhibit some variations, slight variations due to local flexibility or local motion and so on. So the diffraction pattern will lack the fine information within it, right? And the resolution is basically a measure of the level of detail of this reflection pattern in the subsequent electron density map. Now, around 1 angstrom, you will see the structures that will be present. Basically, those will have very high resolution. So that means that in the crystal, they had highly ordered arrangement, which could allow clear visualization in the electron density map. And then for lower resolution structures, which have, let's say, upwards of 3 angstrom, so that can reveal the basic contours of the protein. But if you go at an atomistic, if you try to look at an atomistic level detail, so some of those details will be very murky, kind of like not very accurate in a way, right? A lot of high and low resolution structures, everything in the middle will be present at the PDB database also. And in this plot, basically how the resolution will allow us to particularly capture the electron density that is mentioned. So for example, from 1 to 3 angstrom resolution are given here. And this is basically electron density maps for structures within this resolution, right? So the first three ones are tyrosine 103 from myoglobin. The PTB axis and codes are mentioned here. And then final example is the tyrosine 130 from hemoglobin from this PTB axis and code. So here for the lower resolution structures, you will see the exact kind of shapes or the electron density around these particular atoms for these for this tyrosine. That is very clear, right? The higher you go in this value, you will see that the fine detail is kind of lost in a way, right? So this is basically a representation of how lower value structures for these resolutions will have better resolution in a way. So this one I have mentioned the site where I took the data from. So okay, that is about the resolutions and the PDBs. Now another concept or another kind of numerical sort of values that Professor Komiha went over was regarding the transfer free energy. Now the transfer free energy, it is kind of another way to indicate, let's say, the propensity of certain amino acid residues to be on surface or in a buried location. And what, for example, this related question, what it is asking is that, so the protein has 25 valines and among them 5 are on the surface. Okay. So the rest 20, they will be in a buried location, right? Now you are asked what will be the transfer free energy of valine. Now the transfer free energy was kind of indicated by this formula. So it is minus RT, then ln f of I, where this f of I is the number of the residue I that is present in the interior location divided by the number of the residue I that is present in the exterior location or the surface kind of location, right? So if you implement this formula, so you'll notice that, so minus R is 1.987, then T is temperature, so it is Kelvin, 300 Kelvin here. So R is mentioned here also, universal gas constant 1.987 calorie per mole per Kelvin. So you put that value and then ultimately you do ln of 4 here. So 4 because among 25 valence, 5 are on the surface. So that means 20 will be in the interior position. So it will basically be 20 by 5. So 20 by 5 is 4. So that means this value will give the transfer free energy in terms of calorie per mole. Now, if you calculate this out, you will get minus 826.37. But you have to remember that your answers are asking you in the terms of kilocal per mole. So from calorie per mole to kilocal per mole, you have to divide that value by 1000 and ultimately you will get minus 0.826 kcal per mole, which is basically minus 0.83 kcal per mole, the answer B here. So if you are asked to calculate the transfer free energy, you can sort of refer to this formula. Now another very similar concept Then this transfer free energy is something that we use to calculate the free energy surface of some system. So basically, if you simulate a system or a protein throughout a, let's say, number of time, for example, 200 nanosecond, it could be 1 microsecond or something like that, then based on their different values, for example, if you do some dimensional reduction analysis like principal component analysis, or if you consider, let's say, the RMST value versus the radius of gyration, which is a part of the compactness, indicator of the compactness of the protein. So you can use those type of different values as your collective variables or direction coordinates, and you can try to, let's say, you will get different data points relating to those, right? And then ultimately, if you are trying to see, okay, so for these particular values, around which of these values, let's say, all of the conformations are kind of concentrated in a way. So you can kind of represent that as a term of free energy. And that free energy you basically calculate using something like this. So minus kBT ln ni by n max. So what it is basically saying is that, let's say, in your total conformational ensemble, uh, if you divide that in, let's say, different smaller subgroups, let's say with a bin size of 100, if you divide that in 100 smaller subgroups, bin is basically small interval range for the values that you are trying to calculate it against, right? So in small interval range, you are kind of dividing the entire set of range of value into, let's say, 100 sub range, and then within those ranges, so in which of those bins uh, basically your maximum number of conformations are present. So that will be your n max. And in reference to n max, you can calculate the population for a particular beam, which is indicated here in ni. And then this will give you, let's say, if you consider the n max beam as the most populated. So with respect to that, how much higher or lower number of population is basically present at the other beams, and you can convert that into these using this Boltzmann formula, a free energy value, where your highest populated beam will have an energy of 0, and then every other beam will get some sort of energy associated with that. So that way you can kind of get an entire energy surface representation of this whole conformation ensemble, where you will see that where exactly these values are concentrated and where let's say the other values are present. And then if you are seeing, let's say, some rare event or some transition state that is occurring throughout the simulation processes, you will see some sort of conformations present in those transition states as well, their energy value related to those, and so on. So that is kind of a free energy landscape view of the conformational ensemble. So sort of similar concept here, similar to this transfer free energy, where you also sort of calculate a proportion in a way and you convert that to free energy. The same concept, that is why I kind of just mentioned here. So I think some brief concepts regarding homology modeling, Professor Komiha also went over. So in homology modeling, what does this SCR term stands for? Any idea? So this SCR is part of homology modeling, and this essentially stands for something called structurally conserved regions. So basically an identifier of the conserved regions through these different homologous protein structures. Okay. So yeah, basically that is written here. So homologous protein structures, they have, of course, great conservations compared to the sequences. And then this is evident through the existence of structurally conserved regions. So even in protein families where significant sequence divergence are there, so you will see structurally those are basically very similar. They have different conserved regions. So those are termed as structurally conserved regions, or SCR. So it basically involves comparing two or more homologous protein structures, which is a process influenced by their availability and the degree of divergence as well. And then you can have the ability to deduce structurally equivalent positions among these structures. So that will give you the idea about the conserved regions in these cases, right? I think another concept that Professor Gomiha went over was the idea about residue contact in a protein structure, right? So, for example, here this contact map was provided, let's say. Now, in this contact map, by looking at it, you are asked that, so which of the following residue pairs will form short range contacts? Now, the concept regarding the short range and long range contacts was that in the sequence space, so sequence space is basically, you can think of it this stage of sequence. So from 1, 2, 3, 4, 5, 6, 7, 8, and so on, let's say up to 300, the stage of sequences are present, right? So sequence at position 1, you have certain amino acid, position 2 have certain amino acid, 3 have certain amino acid, and so on. So this is just the stage of sequence and the information for that. So if in the sequence, two amino acids are, let's say, present very at a very close proximity with one another. So that means that one is present at, let's say, sixth position, and then another is present at the ninth position. So these two sequences will be very close in the sequence space, right? So if one amino acid was in the 12th position, another one was in the 15th position, so this will be in a very close position in the sequence space by considering just the sequence, right? Because it is just three residues apart from one another, right? Similarly, if, for example, one amino acid residue is in the sequence, it is in, let's say, 20th position, and then another was in 55 position. So this means that these two residues in the sequence space, they are very far apart, right? However, in the 3D structure, the sequence can basically fold in a way that even though these two different amino acids in the sequence seem to be present very far apart, if we just consider the sequence, one at the 20th position, another at the 55th position, the amino acid chain can fold in a way in the 3D conformation that these two particular points can come at a very close proximity with one another, right? Now, if in a 3D space, in a 3D structure of that protein, if two amino acid residues are in a very close contact, let's say if you are considering with respect to certain threshold, let's say these two amino acid residues in the 3D structure that are present 5 angstrom apart, let's say, right? So that means that they have some sort of contact in the 3D context, right? In the 3D space context. So at the actual structural perspective, these two amino acid residues have contacts. But if those two residues are present in the sequence space very far apart, for example, 20th position and the 55th position, so that means that the type of contact that they are forming, that will be a long range contact. So in the sequence, those are present very far apart, sequence positioning. However, in the 3D space, they fold together to basically come very close. So the contact that will be formed there will be termed as a long range contact. Similarly, if two sequences on the sequence space, they are also very close, for example, at the 5th position and at the 8th position. So in the sequence space, only 3 positions apart, those two residues are. And on the 3D structure perspective also, those two residues are very close together. Let's say within a 4 angstrom distance, their side chain is present, right? So that means that they are also forming a contact. They are also in the sequence space very close positioned together, closely positioned together. So those will be forming a short range contact. So short and long range contact, both of these cases, the 3D structural perspective contact is forming. So on the 3D structure perspective, they have some sort of, they are present very close to one another. But on the sequence space, whether they are present just in adjacent positions or in a very far apart positions. So that is basically what the question is asking here. In this contact map, at every dotted position, you are basically expecting that a 3D contact is formed. So that means in 3D structural perspective, those residues are in contact with one another. Okay, so now the question is asking that, so which of these two residues are forming short range contacts? Now, of course, you have to take a look at whether these two residues that are mentioned here are forming contacts at all or not in the 3D structure perspective. So you can take a look at whether at the junction point dots are present or not. So if the dots are present, that means that in the 3D space they are forming contacts. First, right? So can anyone try to answer this in the chat? What would be the answer to this question? So here, if you look, for example, isoleucine 4 versus GLU 8, first you need to investigate whether they have a dot present between them or not. So here, for example, isoleucine 4 is this one, and then corresponding GLU 8 is this one. So there is a dot present here, right? So that means that these two are forming contact. Then leucine 2 and arginine 6, if you see, so leucine 2 is this position, the one that I'm highlighting, and then arginine 6 seems to be this one. So it is also having a dot, so there is a contact. Arginine 6, isoleucine 7. So if you look at this, arginine 6 is here. Isoleucine 7 is also here, so there is also a dot here. And then serine 10 and then leucine 2. So serine 10 and then leucine 2, if you look at here, so serine 10 is here. Leucine 2 is here. So that is also a dot present between them. So in all of these cases, in 3D space, a contact is forming. Now what you have to ask is basically, let's say among these four pairs, which one is present basically in the sequence space very close proximity. So it seems that arginine 6 and isoleucine 7 is just like one residue apart, right? Just adjacent to one another. So in this case, that will be considered as part of a short range contact. Now, the cutoff for short range to long range, it could be like different considerations might be made there. So for example, you can take like a 3 residue, uh, like 3 position difference cutoff, or let's say 5 position difference cutoff, and so on, to distinguish between short, long, and so on. So in this particular case, the most appropriate answer, of course, because these two are arginine 6 and isoleucine 7 are just adjacent to one another. So you can say that these two will be forming most definitely short range contacts, right? For example, serine 10, leucine 2, they are like 8 positions apart. So even though they have contact, so this will probably be categorized under long range contact. Okay. So I think we will also see some concepts regarding these, like long range order and so on, in the subsequent classes. So yeah, so those were some concepts regarding the classes. I think we can solve maybe one more question. So here, for example, if you are asked, so the accessibility of this residue, which is arginine 74, so the extended and folded states are given like 165 and 90 angstrom square. So the solvent accessibility is being referred to here. So the percentage of accessibility, if you are like given some sort of numeric like this, so you can do calculation using this formula. So accessible surface area in the folded state, and then accessible surface area in the extended state, you can divide that. You can divide the folded by the extended, and then you will get some sort of fraction, which you can try to get the percentage from by multiplying with 100. I think I have missed the times 100 here, but you can get the percentage from that using these sort of formulas, right? So it is 54.55. The number of structures on the PDB ID, so the PDB database, for example, if you're asked, so this is sort of an ever-changing value. So what happened, or how many structures was present like a year before? So of course, a lot of new structures will be added after that. So this is kind of, this value is kind of ever-changing. Uh, so this 2164778, this value is kind of outdated in that sense. We might try to look it up in the actual statistics for the PDB database. But anyway, up to 2024, if we sort of plot how many number of entries in each year that occurred at the basically different years, so annual entries and so on. So you will see that here the number of structures for annual release, and then the total number of entries that are present, and you will see kind of sort of a very steep increase, right? So the more time goes by, the more number of structures are available in the PDB, which is overall good, because uh, you will get a lot of different options if you kind of want to tailor your study in a particular way. So not only the structures for a protein, let's say for example, that protein co-crystallized with different types of ligands will also be available, and you can compare and match those, and uh, what kind of behavior is going on inside those structures. You can look into it. So more options is kind of good in this case, which we will get more of, as the years go by. Okay. So I think today I will just wrap up the class here. Okay. So again, thank you everyone for joining, and then we will of course meet in the next class. Okay. So thank you all. You can please try to join the other TA sessions also. So on Saturdays and on Mondays, those TA sessions are also occurring. And then please carefully follow Professor Bhumiha's lectures, and then again next Friday we'll meet for the next class. Okay. Thank you.